Yeah, I feel forward to that way. I didn't make it hard.
Okay, so the previous session was all on the image processing part of the pipeline. This, as we mentioned, was MIBI specific, and the goal is to take the direct outputs of the MIBI microscope and convert it into something that is cleaned up, processed, and ready to be analyzed. Every part of this step is sort of contained within the talking repo that we talked about, because it's all MIBI specific and deals with a lot of the low-level intrinsic features from the data. We're now going to be transitioning to just general image analysis. So this will be applicable to sort of any of the multiplex imaging or just low throughput imaging approaches that you're considering. The goal is just to learn interesting features from the image data, and these images we're assuming have already been cleaned. So there's a lot of sort of platform specific processing that might happen for fluorescence or IMC or MIDI. The ARC analysis repo, which is what contains all of this, sort of assumes that you've already taken care of it, and now you just want to learn interesting things from the data, now that the data has been cleaned up, to remove whatever sources of platform-specific noise are present there. We'll give an overview of the session. First talk is going to be looking at pixel level clustering. So this is an overview of the Pixie algorithm that we recently published a preprint on, and sort of giving the high level understanding of how it works, what we decided to do, and benchmarking. We'll then have a second talk. Both of these are from Candace, the first author of the Pixie paper, talking about how to actually apply this to your own data. What are some of the considerations when you're going to use Pixie to analyze your images? What are some of the nice features we put in place that we think make this really easy to use, and sort of give you the more application-based session. And the goal is really understand how to use Pixie in general for pixel level analysis of your data. And with that, I will turn it over to Candace.
Okay, hi everyone, I'm Candace, and for the rest of today, I'll be talking about how to use pixel level analysis to analyze your multiplex imaging data. So first, I'm going to give some motivation of why we want to do pixel level analysis at all. Then give an overview of the pixel clustering pipeline in Pixie. So this first part of the talk of today will tell you how to go from like your MIBI images here to your pixel cluster overlay, as represented in the middle there, where each color represents a different pixel cluster. And then tomorrow we'll be talking about how to go from your pixel clusters to your cell clusters, which is the far right image there. I'll talk about some ways that we benchmark the performance of our pipeline. And finally, give some examples of pixel clustering across various biological contexts, as well as different multiplex imaging platforms. At this point, I'll stop for some questions. And then the second session will be about applying pixel clustering to your own data, and some practical tips about how to work with our pipeline. Okay, so first, what does a typical image analysis workflow look like? So after you've applied your images, the first step is to clean up your data. And this was like the whole first session today of how do we get the true biological signal from our data and remove as much noise as possible. The second step of a typical image analysis workflow is to do cell segmentation, which Noah is going to talk a lot more about tomorrow. And that basically just means identifying the cell boundaries in your image. And after you've identified where the cells are, you want to know what the cells are. So that's the cell phenotyping step. And after that, you can do all kinds of cool spatial analysis. And this will be a big session tomorrow. This is really sort of like the meat of imaging. It's like, why do you want to do imaging at all? And finally, we can relate all these features to cool biology. So typically what we do in other multiplex imaging studies, we jump straight into cell level analysis. So after we clean up our data, we'll go straight into cell segmentation and phenotyping. But there's two main issues with this. The first is that the current paradigm for cell clustering is very labor intensive. So this will be sort of the motivation for the talk tomorrow of how we try to make the step less labor intensive. And secondly, information is lost when we analyze images only at the cell level. So today I'll be talking mostly about this. So in our images, we capture a whole bunch of stuff, but we usually only focus on the cells. But the extracellular features can have very important biological functions. So these are just two examples here. So the extracellular matrix has like a growing role in like cancer and other kinds of diseases, and protein aggregates have long been associated with various neurodegenerative diseases. And these are just 2 examples. There's all kinds of stuff that you're capturing in your images, but you're really missing out on if you're only looking at cells. And so here I'm just showing an example of a lymph node image. On the left is a MIBI overlay. The gray are the HH3s, which denotes the nuclei. And on the right is a pixel phenotype map where all the colors correspond to a different pixel cluster. And overlaid on top of the pixel phenotype map are the single cells, which are like the gray circles. So in a lymph node tissue, it looks like the cells are covering, the entire image because the tissue is very dense. However, in other types of tissues, such as in ductal carcinoma tissue or in triple negative breast cancer, we can see that the cells actually don't cover the majority of the cells. And there's a lot of stuff that exists outside of the cellular space. So if we were to actually quantify this, you can see that in DCIS and in TNBC, the majority of the pixels in the image are actually outside of the cellular space. And so when we're doing things at the cell level, we're like completely throwing away all of this information in the image. So we wanted a way to be able to quantify these features. So when thinking about how to do this, you can think about it analogously of how we would normally cluster cells. So I'll be talking a lot more about this tomorrow, but what we typically do at the cell level is we extract our single cells from our images through cell segmentation, and then feed that into some unsupervised clustering algorithm. So all an unsupervised clustering algorithm does is that it groups cells together that are more similar to each other. And then the user can then go in and manually annotate these cells to whatever phenotype they represent. So for instance, we could say like the blue clusters are T cells, the red clusters are B cells, etc. So analogously to how we do this at the cell level, we can also do this at the pixel level. So we can extract single pixels from our images and identify clusters of pixels. So Pixie is the method that we've developed that can do that can achieve the quantitative annotation of both pixel and cell level features. Today I'll just be talking about the pixel part, and tomorrow will be the cell part. So I'll say, so Pixie is a name that we settled on, but Mike had a lot of great suggestions that we unfortunately didn't use. Pixel Me Elmo is his favorite. Pixel Sowers was another one.
I still think this was a lost opportunity, but I've let it go.
So we went with Pixie. And so today I'll be showing how we can use Pixie for the quantitative reproducible annotation of features not captured by traditional segmentation, and also applying Pixie across a variety of different multiplexed imaging platforms. And then tomorrow I'll be talking about how we can use the pixel cluster to improve our cell data. Oh, and this method was recently put up on Varra archive, so if you have any more detailed questions, feel free to look at the paper. Okay, so as a proof of concept, and to show what pixel clustering can really do, as well as benchmark its performance, we use a data set of lymph nodes. And so one of the reasons we chose lymph nodes is that they're very structured tissues, so we're able to assess how well our pixel clustering does by looking at like the general structure of the tissue. So for instance, here in this image, the red is CD4, which denotes the T cell zone. The blue is CD20, which denotes B cells, and that's the B cell zone. And in green is CD21, which is follicular dendritic cells, so that's usually the germinal center. So as you can see here, it's very structured tissue. Lymph nodes also contain a lot of cells that are difficult to segment, such as like follicular dendritic cells or macrophages. And so using pixel clustering, we're able to sort of look at a lot of those kinds of features. So this is what the pixel cluster overlay looks like of that image. Here, every color corresponds to a different pixel cluster, and hopefully you can appreciate that we're able to capture the major structural features of the tissue. And so I'm going to be showing you a lot of these heat maps throughout the presentation, and they just tell you what the pixel clusters that you're actually looking at are. So in all these heat maps, the rows are the pixel clusters, and the colors here correspond to the actual image. And the columns are the different markers in your panel. And then the colors in the heat map just tell you like which pixel clusters are expressing which protein. So just moving into some examples here, for example, if you look at this brown pixel cluster, it's a CD209 to a 6 pixel cluster. And if we look at that in comparison with the actual MIBI images, you can see that the signal lines up really well. Here's another example of CD8. Again, lines up very well with the actual MIBI image. Okay, so this is the full clustering pipeline in Pixie. So Pixie includes not only just the clustering steps, but all of the pre-processing steps as well. So I'm going to be talking about each of these steps in a little bit more detail. Before going into that, we wanted to benchmark the performance of our pipeline. So when we were developing this pipeline, I showed you in the previous slides are like the nice set of pre-processing steps we decided on, but we tried so many more different pre-processing steps and also different parameters for each of those steps. So we wanted a quantitative way to be able to assess what was actually performing the best. And so many high-dimensional data analysis techniques are stochastic, so that means that they are inherently random. So if you take the same data and you run it through the same algorithm multiple times with a different random initialization, you'll get back different results. And so like UMAP, TISNI, these are all actually stochastic. So we reason that good clustering results that reflect true biological phenotypes should be reproducible across different random initializations. So we use this as a way to assess how well a particular set of pre-processing steps was doing. So this is an example here of another lymph node image ran through the pixel clustering pipeline 5 different times. And so hopefully it looks very, very similar to you. If you stare very, very closely, you will see small differences. But the fact that it is very reproducible is a good thing. And here are the heat maps corresponding to each of those 5 replicates. Again, each of the rows here is a pixel cluster, each column is a marker. So in an ideal case, we could assess this idea of like stochasticity by simply assessing how often a particular pixel is assigned to the same feature. However, in reality, unsupervised clustering algorithms rely on this manual annotation set. So for instance, like in replicate 1, the T cell cluster might be like cluster number 1, but in replicate 2, the T cell cluster might be like cluster number 2. And so you have to go in and manually annotate each cluster number with its phenotype. So that's what I did here, which is why the colors like line up so nicely. But you can imagine this is infeasible for a very large number of tests. So we tested a whole bunch of different pre-processing steps, a whole bunch of different parameters. You map into 5 replicates for each one. So having to go through and manually annotate every single one would take a very, very long time. So we defined this quantitative metric to be able to capture this idea of like how reproducible is it across different random initializations. And so we call this score the cluster consistency score. And it can be roughly interpreted as the number of different clusters that a given pixel was assigned to across replicates. And so the actual calculation for this score is a little bit complex, so I'm not going to go into the details here, but the bioRxiv paper has the entire algorithm laid out if you are interested. So after we generate these scores for every single pixel, we can overlay them onto the original image to get a good idea of how well a pixel cluster was actually doing. So in all of these pixels, or all of these cluster consistency score overlays, a lower score is good. That means that pixel was almost always clustered with the same pixel across different replicates. But a very high score is bad. That means that it was kind of sometimes in a lot of different clusters with a lot of different pixels. Okay, so now that we have a quantitative metric for assessing reproducibility, we can use this metric to assess each step in our pipeline. So the first step here is the Gaussian blur, which I think we talked a little bit about already today. So I like to think about like Gaussian blur. The reason for doing a Gaussian blur is similar to like why we compensate for dropouts in single cell RNA data. So in RNA-seq, in single cell RNA-seq, you're not actually capturing all the transcripts in your single cells. Similarly here, maybe it's not actually capturing like all of the protein expression in the tissue. So you can think about Gaussian blurring as like smoothing the data, like compensating for dropouts. So here I've assessed 4 different sigmas for the Gaussian kernel in the Gaussian blur. So on the very left here is no blur at all, and it looks very like salt and pepper, and it looks like it's quite noisy. And as you increase the sigma, the features get sort of more smooth. And so for all of these different sigmas, I calculated the cluster consistency score. And again, like a low score is good, high score is bad. So hopefully you can appreciate that sigma 2 was like the best value for this data set. And we tested this across a variety of different data sets in the lab. Sigma 2 seems to work well, but this may be a parameter that you can like change in your own data. Okay, so the next step of the pipeline is this pixel normalization step. And so in this step, what we do is divide each pixel by its total expression. So what we end up with is that each pixel sums up to 1. So why do we do this? So the reason behind this step is that when we're doing phenotyping, what we're truly interested in is the ratio of the phenotypic markers. So the absolute intensity of the pixels can be different for a variety of reasons. For example, like biological, there's downregulation of receptors upon activation, or technical, which we've talked a lot about already today, such as drift and instrument sensitivity. So because of these reasons, the absolute intensity of the pixels can be different, but they may represent the same phenotype. So for instance, if you have a T cell that after activation downregulates CD3, a very bright CD3 cell, a very dim CD3 cell, they're both T cells. You want to identify them as T cells. But if we leave out this step, then we get like a very high expressing CD3 cluster and a very low expressing CD3 cluster. And yeah, so without this step, we see that there's one dominating pixel cluster that is clearly defined as outlined in the box up there on the right. So just by looking at the expression of that cluster, we're not able to assign a phenotype to that cluster. And it takes up a lot of the pixels in your data set. So on the bottom are just 3 images with the pixel clusters overlaid. That gray cluster is the unassigned cluster. So without this pre-processing step, it looks like it's very hard to actually assign phenotypes to your image. And in comparison, these are the same images with the step added. So as you can see in the heat map, each pixel cluster was able to be assigned to an actual phenotype. Okay, the next step in the pipeline is 99.9 marker normalization step, which means that we divide each marker by its 99.9 percentile. And the reason for this step is that we don't want very bright markers to drive the clustering. And without this step, we see, again, many poorly defined clusters. In this case, it looks like the clusters are expressing a lot of different proteins. So it's hard for us to make, to give it an annotation. And if we assess the cluster consistency score, we can see that it gets really, really bad without this 99.9 normalization step. And if we quantitatively compare these 3 different scenarios where we use all the pre-processing steps, no pixel normalization or no 99.9% normalization, including all the pre-processing steps, does significantly better. Okay, so those are all the pre-processing steps in our pipeline that we settled on. And after we've done all that, now we can actually go into the actual clustering. So to do the clustering, we use an algorithm called the self-organizing map, or a SOM. And there are a lot of different unsupervised clustering algorithms out there, but we chose this on because it is really fast. It is really important when we're talking about clustering pixels because there are millions to billions of pixels in your images. So what we typically do is that we cluster all of our pixels into a larger number of clusters. We call this over clustering. And the reason we do this is that we found that it is better able to capture really rare phenotypes if you first over cluster. And so after the over clustering step, we then do a meta clustering step where you're then combining your 100 or 200 pixel clusters into a smaller set of clusters that are more biologically relevant. And finally, then we can manually adjust these meta clusters to make sure they're actually lining up with what we would expect to see. And so to do this, we have developed a graphical user interface, or GUI, that is part of our pipeline in ARC. And I'll be talking about that a lot more later. And so for the middle step here of the hierarchical clustering step, you have to tell the algorithm how many hierarchical clusters you want. And there have been a lot of algorithms developed that like algorithmically, anyway, help you the number of clusters that you want to use for your meta clustering. However, we found that like assessing the heat map is actually a very good way of choosing the right number of meta clusters. And previously, I think I've been difficult to do this because there hasn't been a good tool available to really interact with your clustering results. In our lab, what we used to do is we just like print out a giant heat map on a piece of paper and just like highlight clusters and try to do it that way. So this new tool we think has really helped with this process. And I'll be talking more about that later. Okay. So another important consideration is that imaging data sets are getting very, very large, right? So imaging data sets now in our lab are on the order of like billions of pixels, whereas in like SciTalk or like single cell RNA-seq is definitely like thousands to millions range. And so it's infeasible to read all of these pixels into your computer's memory at the same time. And so we hypothesized that we could train the SOM, the self-organizing map, using just a subset of all the pixels, and that would give the algorithm enough information to still make accurate computations. So to show that, I used a data set that was recently, or not recently, last year put on BioArchive. And so these are just two example FOVs from that data set. These are the MIBI overlays, and these are the pixel cluster overlays using a SOM trained with all of the pixels. So this data set was around 800 million pixels. So I was able to train a SOM using all the pixels, but it took a significant amount of computing power. I think I used a Google Cloud instance and jacked up the RAM to like 800 or 900, and it still took multiple hours. So it was not feasible to do for data sets moving forward. So what I did was I subsampled the pixels randomly to just 10% of all the pixels and trained the SOM using just 10%. Hopefully you can appreciate that the phenotype was very similar. And if I were to quantify this, we can see that the correlation between all the pixels or the 10% of the pixels is very high. Okay, so that was like the full pipeline, and now I'll talk a little bit more about how we use pixel clustering across different biological contexts and across different imaging technologies. In this first example, we use Pixie to map the full neural landscape in human brain. So in brain MIBI images, it has historically been very difficult to use traditional segmentation methods or object detection methods because neurons are so weirdly shaped and have like weird spatial conformations. So you can have like neurons coming at you in like the Z direction and also like intertwining with other cells. So it is traditionally very difficult to identify these objects. So this is a pixel cluster overlay of a stitched human cerebellum. And I don't really know anything about the human brain, but hopefully you can appreciate that you're able to capture like the structure of the tissue. Wow. And so this is the heat map that corresponds to the different pixel clusters. And just moving into one example, we can see that the signal of NEFH, which marks gray matter axon, lines up really nicely with the MIBI image. So that's this like red pixel cluster here, so there's like red squiggly lines. It is reflective of the actual MIBI image. And it's like such a weirdly shaped thing that would probably, or like definitely not be captured using our like current cell segmentation method. Okay. In a second example, we applied 60 to a publicly available Codex data set of colorectal cancer. So Codex is another multiplex imaging technology that uses fluorescence tagged antibodies instead of mass tags, which is what we use. And so a big confounder in fluorescence methods is that you get a lot of tissue autofluorescence. And so in this data set, what I did was I included images of empty cycles. And what that means is that these are images that don't actually have it, that aren't actually looking at protein expression, but are just quantifying the autofluorescence in your tissue. And so by including images of just the autofluorescence, I was able to pull out this like background pixel cluster, which is denoted in black here, and overlaid onto like the pixel cluster overlay. You can see that it does sort of capture the background well. And in addition, we're able to capture like a lot of the biological features we would expect to see, SMA and CD20 machines there. Okay, so next, we also applied Pixie to a MALDI data set. And so in contrast to both MIBI and Codex, MALDI is a label-free technology, which means that there are no antibodies used to target specific proteins. So MALDI can be used to map the glycan abundance in our tissue. So I'm not going to like try to explain the details of MALDI because I'm not good at that. But the important point is that the output of MALDI are these single channel images where each channel here corresponds to a different glycan. So these are some example images. So the intensity of the image signifies the abundance of the glycan at that location, and the little cartoons on like the right top corner indicate like the glycan structure. So we applied Pixie in this publicly available adenocarcinoma data set, and these are the pixel clusters that resulted from it. And what's pretty cool is that if you compare this with the actual with the H&E that was in the original publication, we can see that the pixel clusters nicely sort of lined up with the histological features that they identified in the original publication. Further, if we look at the actual glycans that were defining these pixel clusters, they lined up nicely with the manually annotated glycans that were talked about in the original publication. So this was an automated way of picking out these glycans that were specific to these different histological regions. And yeah, so in addition to confirming what we saw in the original publication of which glycans are expressed where, using pixel clustering, we're able to identify other co-expressing glycans. So for instance here, so again, each row here is a pixel cluster, each column here, instead of proteins, these are all different glycans. So the names look a little crazy, but that's what they are. And so, yeah, so as an example, this is the region in the previous image, which is the necrotic region. So it's that like blue, purple cluster in the middle. And if we look at the glycans that are defining this region, it does include the glycans that were mentioned in the original paper. But upon reviewing these glycans a little more closely, we can see what's kind of cool is that they all come from a similar family. And I really, hey, I'm so sorry, I meant to put your name up here last night, but hey, in our lab, the driver of figuring this out. And so, yeah, so it's kind of cool that it comes from similar families. So if we just focus on these 4 glycans here, we can see, oops, wrong way. We can see that they all kind of have a similar structure and really differ in this, like the top region as indicated in the red box. And looking at some of the other glycans that were also in the cluster, they again have similar structure but have various modifications added on, such as silation or a fucosylation. And so the fact that all of these glycans are very structurally similar, they give us a hint of like a common origin of some glycan that these were all generated from, and potential biological mechanisms of like which modifications are added in like which regions of the tissue. Okay, so finally, I'll share a small vignette about when we use pixel clustering to investigate the transition from ductal carcinoma in situ, or DCIS, to invasive breast cancer, or IBC. So this was published earlier this year in Cell. And so DCIS is a non-obligate precursor to IBC, meaning that not all women who have DCIS will progress to IBC. And why some women progress and some don't, the reasons behind that are like relatively unknown. And so the normal breast tissue is defined by this like green myoepithelial layer that surrounds the ductal cells in green there. And in DCIS, this layer becomes very, very thin and stretched out. And in IBC, it completely breaks apart, allowing the tumor cells to invade other places. And so this myoepithelial layer is a very important histological region in understanding why DCIS progresses to IBC. But that layer is pretty difficult to analyze in our images because it's such a small histological region. And also the cells become so thin and stretched out that traditional cell segmentation methodologies aren't able to capture those cells well. And so we wanted to be able to quantify the features in this like small myoepithelial layer. So to do that, we applied a myoepithelial mask on all our images to only look at the pixels within the myoepithelium. And cluster just the pixels within that layer. So these are the pixel clusters that resulted from that. And on the right here, I'm just showing some pixel cluster overlays. The black region denotes the myoepithelial layer. And hopefully you appreciate that the pixel clusters line up nicely with the actual protein expression. So for instance, like the E tag is this like yellow, and we can see that it lines up well with the green signal here. So in this paper, we also built a random forest classifier to try to identify DCIS features that are correlated with recurrent outcomes. So we separated our patients into 2 groups, progressors and non-progressors, and then built this random forest classifier using a total of 433 different MIBI features. So these features included like cell type prevalences and like various spatial metrics that we pulled from our data, which we'll talk about tomorrow, including the pixel cluster frequencies that were identified in the previous slide. So this is the result of the model. And on the left is just the AUC curve, and on the right, we're showing the Gini importance, which is a measure of how important each feature was for the model's performance. And what's pretty cool is that like the #1 important feature for classification was the frequency of this ECAD here in pixel cluster. This is cool because the myoepithelial layer is already a very small histological region of the tissue, and the ECAD pixel cluster is an even smaller percentage of that. So it's such a small percentage of the actual images, but it has some sort of cool biological significance. So using pixel clustering, we're able to like pull this feature out from it. Yeah, so in summary, there's important information that is captured outside the cellular space. And Pixie is a complete pipeline for the annotation of pixel-level phenotypes in multiplex imaging data. And Pixie can be used to phenotype acellular objects and cells across various biological contexts and also imaging platforms. Okay, so this one I'll stop for questions.
So have you ever run it with including control tissues, and would that like skew or improve results?
Yeah, that's a good question. So actually in this SciMac paper, I don't know what SciMac stands for, but it's a paper recently put up on laboratory investigation. It's actually published. It was a tissue microarray, including a bunch of control tissues, as well as different carcinomas and sarcomas and all those kinds of things. And we ran pixel clustering on that entire data set, including all the control tissues and the disease, and we were able to like pull out the phenotypes that we would expect. As to whether or not it would improve it or not, I think it just depends on like which phenotypes are in the images that you actually feed into the algorithm. So it may help because it tells the model like what a T cell pixel is, what like a B cell pixel is, even if those are not in the carcinoma or sarcomas or whatnot. But I think it really depends on the question of whether or not we want to include control tissues in the history.
So in some parts you mentioned when the data set is large, you selected a subset of the pixels. Can you explain how you determine which pixel you select? Is it random, or is it sort of downsize the image, like reduce your resolution from by twofold or fourfold or something like that?
Yeah, that's a good question. So we just ran an example of people on an FOV basis. And that is, I mean, doing the random selection is definitely the simplest way of doing it. And so we thought about doing like fancy, more like density dependent downsampling, but because the random sampling seems like it did well, we didn't try to go down those other routes. I think that there's so many pixels that like just a random sample is able to capture the like full spectrum. Whereas if you have like thousands of cells and you're doing like random sampling of just 1,000 or so cells, then you might not be able to capture like the full spectrum of phenotypes. But I think in these data sets, like billions of pixels, even if you're taking like 10% of that, it's still a billion. It's still so many people that I think we're still able to like capture that full range of phenotypes.
So does that mean only those 10% of the pixel that you actually selected, that you assigned a phenotype to them, and the rest, like 90%, you didn't really assign any based on sort of interpolation or anything? Okay.
No, sorry. Yeah, so we use the 10% just to train the self-organizing math. You can think it's like basically training a model, and once you have the trained weight, then you apply it to the rest of your data. So in the end, each pixel is assigned to a phenotype, but like which phenotype is assigned to, that part is determined only by the 10%.
Okay, it's like supervised learning?
Kind of, because it's just that you're training a model and then applying that model to unseen data. I wouldn't say supervised, we still don't know like the label of the actual data. Yeah, that makes sense. So like you don't have like in a normal machine learning model, you would have like a training set and like a test set, and only train the model with train data and then apply it to the test set. It's kind of similar where you're only training on like a subset of the data and then applying it to the rest of your data afterwards.
How about the normalization part?
Yeah, that's a good question. We first pre-normalize them.
What method do you use for normalization? The max normalization, the normalization, there are different ways of normalizing?
Yeah, so we just do the really two things. Well, I mean, this was stupid to try to go all the way back, but like the pixel normalization we divide each pixel by total, so that is independent, and then a 99.9% normalization step. So we calculate those 99.9% normalization factors across the entire data set first. And that's how we normalize those two steps.
Okay, thank you.
Yeah,
It's great work.
yeah, Lauren.
So like in my head, it's just this pixel normalization step here. So I can imagine that it works with MIBI data really good after you apply the blurring, because then there is essentially not much noise. But when there would be like high frequency noise, you would just upscale this, and like your pixel clustering maps would look like grainy, noisy, or also, right? So, and in the codex data set, there is lots of noise, but you included the autofluorescent channel. So after the normalization step, all the autofluorescence will be like high, well, the channel will have a peak there. So I'm thinking about the limitations. So this wouldn't work for something like codex without autofluorescence probably, or for like other imaging modalities that are super noisy and there's no noise channel, right? Or would it? That's the question. Like, where would it break?
Yeah, that's a good question. I think it depends on the level of noise in your data, and I think by looking at the actual images, would be a good way to assess that. And I think a lot of people do use like a Gaussian blur, like a huge black denoising.
Yeah.
So I feel like, yeah, I think it's specific to the kind of type of data you're working with. So I think with the MIBI data, we actually didn't Gaussian blur that at all, I believe, because it's not as high resolution. Where's the word for that? It's not as high resolution as MIBI data. So in that case, we didn't do a Gaussian blur. So I think the best case to do is for your own data is to just try a few different Gaussian blurs, look at the actual data and see like what the noise level is. And so if there's a lot of noise in your data, when you overlay the pixel cluster overlay, it'll look very obvious. It like looks super like crazy.
Yeah.
There's salt and pepper everywhere. I think if that's the case, then we can think about like other methods of like trying to normalize. We tried a whole bunch of different like ways to like normalize our data to perhaps one of the other things that we tried would work better for different kinds of data sets. But for all the stuff we tried, like these seem to work well.
I think, so the other thing is that, you know, that I think like the intent with this is that the data, you know, that like Pixie, I don't think is really intended to be a means of removing systematic background. Like the sort of the underlying assumption is that the data that's going in is like, can be viewed as ground truth,
Yeah.
right? And I think in the case of the stuff with Codex, really like if you look at that image, what you can see that it's doing is it's really just honing in on slide, which is where there's no other signal in any of the other channels. So it's not, so because all the pixels are getting like sort of a categorical call and they can't be proportionally, right? So it's not like it's not like a weighted sum of multiple different, like, you know, sort of orthogonal variables. So I think like to your point, you can't really this can't really be used as a means of removing something like autofluorescence, which is an additive signal on top of the real. Yeah.
And that's true for everything in the ARC pipeline.
Yeah.
The reason we say we have Toffee as a separate thing is because we're assuming that wherever your images come from, when they're coming into ARC, you have already dealt with the systematic sources of noise in a platform specific way, whether that's autofluorescence correction, whether it's, you know, our Rosetta approach, whatever people have decided on is the best way to get rid of the platform specific issues. We're now deciding that whatever is left in the image, we are like comfortable with assessing as true signal.
So if you have systematic, you know, signal in there, it will confound the classification. So it is something to remember that it's, you know, those things need to be sussed out before they go into any of this.
Can you talk about setting the number of individual clusters and the final number of meta clusters, and like what does that depend on? You know, data set size, data set complexity, different tissue type. How do you think about the right defaults?
Yeah, so for the over clustering step, we typically would just try 100 just to look. I think it will depend on how many markers you have in your panel. And I'll talk about this a little bit later, but usually for all these clustering steps, we only include the phenotypic markers. So it's more like how many phenotypic markers you have in your panel, how many phenotypes do you expect to see in your tissue, and like how rare are those as well. So for instance, like in some of our data sets, like there's a very small number of Tregs, but like FOXP3 is in the panel. So we would expect to see that FOXP3 positive pixel cluster. But sometimes we won't see that until we increase the number of metaclusters, over clusters to a large number. So it is, I think, data set dependent, but in our case, like 100 or 200 have usually been like our go-tos. And then the final number of metaclusters, so you can set, you know, the initial K for the metaclustering to be like 15 or 20, depending on how many things you expect to see. But really, it's like when you're assessing that final heat map, that will give you the best idea of how many metaclusters are actually in your data, because you in the heat map, you want each metacluster to be defined by, you know, 1 or 2 markers or whatever your phenotype is. So yeah, so looking at that heat map is really the best way to choose. I think you can get, you know, as granular as you want. Sometimes, like if you're looking at just like the macrophage cluster, right, you just like anything that's CD68 positive, you're just like lump in there. But if you want to get like super granular, you can like separate out the 68 positive and like the 206, 209 positive. There's a lot of like combinations of things that are possible. So it just like depends on your cluster as well.
So you talked about, you know, pixel clusters to identify these kind of acellular or hard to segment features. What is the subsequent step to actually use that? To generate a mask to go for those objects? Is it like counting the number of them in images? What are some ways you could take microglial projections or like tau things and then actually use that for your analysis?
Yeah, that's a good question. I think, again, like, not to sound like I don't know the answer to all these questions, but I think it's very data set specific. So for instance, in the brain example that I showed, Sunya and Sean's lab generated those images, and I believe she's doing some sort of neighborhood analysis, still a work in progress, to kind of look at what other pixel clusters, if you're looking at like the axon pixel cluster like I showed, like what other pixel clusters are in its surrounding. Yeah, you can think about doing all of these, or using the pixel clusters as like another mask for object detection if like you want to detect a certain object that's defined by a pixel cluster and find like a larger contiguous region, like this is the object. We can also think about ways of doing that. Yeah, I think the possibilities are endless. What you could do. Yeah.
What about gross tissue structure, like large scale features? Do you think pixel clustering can be used for that?
Yeah, so for example, in like a lymph node, like a T cell zone or the B cells or a germinal cell or something like that. Yeah, I think you can. I think it's like how granular you want to go. Like for instance, in the lymph node examples that I showed, the T cell zone was composed of not just T cells, but like dendritic cells and macrophages, all kinds of stuff that's also in there. So if you're only interested in getting like larger areas of regions, you may have to mask based on like multiple pixel clusters or something like that. But we haven't really tried doing like larger regions.
Why did the sigma level have an impact on the cluster consistency score?
Yeah, I mean, that's a good question. I think of the cluster consistency score as like a metric for how like well the pipeline is doing, right? So like for a very low sigma, there's a lot of noise, right? So like it's not very clear what the separation of the data actually is. So it's like a lot of pixels may look like you're expressing like a whole bunch of different stuff. And so it's very actually hard to cluster those pixels because like sometimes one is random initialization will be clustered with like some other stuff, and another random initialization will be clustered with something different. And that's just because the data itself is very hard to cluster because it's not very, each pixel is not like clearly defined. And so I think increasing the sigma of the Gaussian blur is able to sort of pull out what the true expression of that pixel should be. And that's why like it kind of helps with like, the performance of the pipeline because it's making it like easier to classify.
Awesome, thanks.
Oh. Okay, well, I'm not done yet. I have a whole other. Well, let's get there. Cool. Maybe I should just hit the slide that we've already made. Okay. Okay, so the second part of the session will be a more practical session about how to apply pixel clustering to your own data.
I have a seizure.
It's just the TV, it's not showing up on.
Okay, cool. I was gonna say, no, look at me, but please don't do that, because that will stress me out. Okay, so how do we apply pixel clustering to your own data? So the first step, which we found very helpful in our lab, is to first manually curate a test set of FOVs. Like we mentioned before, running the entire data set through the whole pipeline can take a very long time. So when you're trying to choose like which parameters to use, and just to like get a general idea of how well the pixel clustering is doing, you probably don't want to sit there and wait for like 5 hours while it runs through your entire data. So choosing a test set of FOVs is a good idea. And so you can randomly choose these if you think that like your data set is like relatively homogeneous, and if you just randomly pick FOVs, you get a good representation of your data. But if you have like different kinds of tissue in your data, like tumor versus like immune, something like that, you might want to manually pick a set that you think is like representative of your full data set. So we found that maybe like 20 to 30 test FOVs, depending on the size of your images and like the total size of your data set. But like 20 to 30 would be a good place to start. The next step is to decide which markers you want to include. So you typically want to include the phenotypic markers, for example, like CD3, CD20, CD68, these things that like really define your cell type. And exclude functional markers like Ki67 and so on and so forth. And so the reason for this is that functional markers can be expressed on a whole bunch of different cells, right? And so the combinatorial possibilities of all your functional markers on all your different cells is huge, right? There's a lot of different possibilities for that. And so trying to include all those markers into one pixel clustering run just gets pretty crazy. And so typically what we do is first use the phenotypic markers to identify your cell type, and then do like functional characterization. So the pipeline for running Pixie is available on ArcAnalysis, and there's a GUI included in the notebook to manually adjust the clusters, which I know I've mentioned many times now, but I will finally be talking about that more. And then you can assess your pixel cluster overlays with the actual multiplex imaging images using whatever software you want. The Parker Institute for Cancer Immunotherapy, or PICE, has developed a tool called Mantis that we like to use. The main feature of Mantis is that you can view multi-channel overlays and then overlay your pixel clusters on top of that and like toggle them on and off to see how well your pixel clusters match up with the expression. And there's going to be a whole talk on that tomorrow, so hopefully it'll clarify some questions there. And the most important thing here is to iterate. So you can first do a round of clustering, then look at your pixel cluster overlays, kind of see how well it did, and then change your parameters and just iterate through this process a few times. Okay, so this is what the GUI for adjusting the clusters and annotations looks like. And so when we were first developing this method, like I said, like all unsupervised clustering algorithms rely on this manual annotation step. And it's very annoying to do this like manually. And so this GUI hopefully makes it a lot easier. And so the way this GUI is like all of this first like massive heat map here, these are the 100 clusters that we have, the over clustering stuff. So each column is one of the 100, and then each row here is one of the markers in your panel. And then on the right here are all of your meta clusters. And so those are basically like the combinations of the 100 clusters here. And the colors on the bottom here indicate which meta cluster these clusters belong to. So for instance, like these clusters here are all combined to make that first complement. Okay, so this is actually what the like use of the GUI looks like. So they're all available on Arc, and this is what, after you go through all the clustering, this is available, or this shows up 100 clusters, meta clusters. So if you want to assign or move the assignment of one of your clusters to a new meta cluster, what you do is you like figure out which one you want to do. You click on it, it's selected. Then you can right click on the meta cluster you want to move it to. So that one was moved here. And then both heat maps will update automatically. And then if you want to create a new meta cluster, same thing, you identify which one you want to click on or move, or create a new meta cluster, click on it, and then click new meta cluster, and it pops out as a new meta cluster over there. And then finally, you can annotate the meta cluster by looking at the heat map. So for instance, that new meta cluster I created looked like it was CD45 only, so we can annotate that as CD45 only. Click on the next one you want to annotate and give it whatever annotation you want. Yeah, so hopefully this makes it a lot easier to annotate your clusters. Like I mentioned, Mantis is the software we like to use for assessing the pixel clusters. So you can look at different marker combinations of markers in Mantis, and you can choose which markers you want to look at here and like different colors. And overlay the pixel clusters on top of that. Here I'm showing it side by side, but it'll show up on top of it, and you can on the right toggle which cluster you want to look at on and off to just look at individual. Okay, so once you're happy with the clusters, then you can run it on your full data set. And so if necessary that you, like I mentioned, want to subsample the percentage of pixels used to train this on. And you may need to iterate a few times of like how much you want to subsample. Yeah, it really depends. Like I think on a regular laptop, you might have to be subsampling quite a bit. But if your lab has access to like a core or use one of the cloud services, you may be able to go a little bit higher with how much you sample. In our experience, 10 to 100 million pixels actually is the start to lower, like 10 is already usually perfectly fine. And then afterwards, you can generate these nice pixel phenotype maps that correlate or that correspond to the different pixel clusters you have identified. And that's it. Yes, Ben.
This is super awesome. But I was just wondering, you were showing earlier that you had multiple different data sets and you wanted to give the same cluster to each like group. So like you said, you would have like T cells in cluster 1 and then T cells in cluster 7 in the next one. Is there a way to like move these presets so that like T cell, like you can always have T cells be the same cluster in every time?
Yeah, so that would be great because then we wouldn't need the cluster consistencies for it all, right? We can just like say like this pixel is a T cell in this replicate and also the other replicate. I think unsupervised clustering algorithms don't do that. There are a variety of other supervised algorithms that are now being developed that can do that. The output is that this pixel is a T cell, this pixel is a T cell, as opposed to like the actual number. But I think the nature of unsupervised clustering is that it just basically clusters.
Yeah.
Did you find any subcellular pixel clusters?
Ooh, that's a good question. I don't know if we have any markers that are trying to look at subcellular structures. I guess like in the brain data, I don't know what like most of the markers are. I wonder if some of those are subcellular, but I'm not actually sure. I think in most of our projects we haven't been interested in like a question where we're looking at subcellular.
Oh, FOXP3.
Oh, yes. FOXP3.
Yeah, so we often find membrane versus nuclear pixel clusters.
Yeah.
So, you know, the T cell, a lot of information that it's a T cell comes from the CD45, CD3 pixel cluster, which is like almost exclusively at the membrane. So in that sense, it's subcellular. And then similar for nuclear markers like FOXP3, those we can be restricted.
Yes.
Yeah, what about in addition to the subcellular question, like with the staining method and looking at like RNA probes? Is that something that you could apply pixel clustering to?
Probably yes. We have not done so yet. I think well, in my specific instance of using Cami, I'm like basic. So for those who don't know, Cami is a way to use to use RNA and DNA inside your hybridization on the MIBI. So you want to look at RNA and DNA and protein in the same section. And so Suzune, who used to be a postdoc at Gary Nolan's lab, published a paper on Immunity earlier this year. So in both Suzune's paper and in my current one, we're looking at viral HIV, viral RNA, DNA inside of cells. So those are, well, I don't want to get into philosophy. It's usually on CD4 cells. I could argue there are a whole bunch of other crap that people just don't appreciate. And so I think like my thought is to more look at just the presence of it, like whether or not that dot is even there in that particular cell type. But I don't think in that situation it'd be even necessary to do pixel clustering because it's just the presence. You could just binarize it in my head. Obviously, I haven't. Well, it's not obvious, but I haven't done that yet. And so I don't know if it'd be necessary, but I feel from like a purely like data standpoint, it's just another channel, so I don't see like why not. You know?
I mean, the one place that might be that might end up being useful is if, if we get to the point, where we're doing combinations of metal channels to identify, transcripts or DNA copy variants, then you might be able to do that to kind of flesh it out. But I agree with you. If it's one metal, if it's like a single metal per transcript. I think the other thing is too, is that you probably don't want to include them with the regular pixel clustering because you don't want to, if it's on top of a protein signal, right? I don't think you really necessarily want to have that as a distinct cluster, right? It's
I
sort
think
of,
I would think of it also almost like the functional markers. It's like you could.
Yeah.
look at it faster.
What are the limitations of the Pixie method and how do you address it?
Limitation. None. I think like my method, I think it's very dependent on the data that you're feeding into it. So it's like, it's not as much as like we like want it to be like magic and like have these beautiful pixel clusters. I think it really depends on like the data that you're feeding in. So it's not gonna like magically clean up your data for you and do that kind of thing. So I think that's why the sort of the iterative process is also so important because you might like look at the heat map, and you're like, oh, look, it's so discrete, beautiful. But then if you look at the image, it might look super noisy, and it's just a captured completely noise. So I think that, yeah, I mean, the limitation is that it really relies on the quality of your input data, I think, to get good pixel clusters. And yeah, that's just the way it is.
Have you looked at any of the multiplex fish RNA methods? Like
Multiplexed,
Wormfish, Deepfish, Vision.
No, but that would be cool, but no, I.
All right, thank you, Candace. Okay, so thank you everyone for sticking with us for day 2. Just going to give a brief overview of what happened and what's to come. First, we talked about the processing MIBI data, Rosetta, normalization, all that good stuff to take the output of the commercial instrument and make it basically ready for ARC. We then heard from Varra, really cool talk on machine learning for alignment and spatial factorization. We just heard from Candace on the latest and greatest in pixel level analysis. Tomorrow, we'll be covering in the first session cell level analysis. So once you've done all this pixel stuff, what can you do with actual cells? We then have a guest lecture from MIBI site at Lehigh talking about their graphical interface for actually using the HE image directly to identify which regions you want to acquire on the MIBI. And then we have our final session on spatial analysis. As you mentioned, there are pre-recorded talks that kind of go more into the details of how to actually run these algorithms. All of the stuff we talked about this morning in Toffee, there are sort of guidelines and videos to walk you through how to set up Toffee. All the stuff we kind of glossed over and said it just works. This explains how it works. And there's also a talk specifically on setting up ARC. Today we heard the first talk from Candace on software that uses the ARC GitHub, which is completely separate from Toffee. ARC is all the general spatial tools that any lab can use. Toffee is the one that ships to MIBI. So there's one on how to get that set up on your laptop. After lunch today, we're having our first set of computational office hours. So this is help setting up the GitHubs. If you have any general questions, troubleshooting, installation, all that stuff, these are the same wonderful folks who have made the pre-recorded talks, help guide you through. So I would recommend taking a look at the talks, coming to office hours to get maximum help for getting all this stuff running, especially if you're maybe less comfortable with computational analysis. And so to get you excited for the office hours this afternoon, we have our computational team here who will be introducing themselves and giving you a little primer. So first up, for all of you up, you can go through it.
Yes.
Hi, everyone. My name is Cami, and like everyone has mentioned, we are going to be hosting computational office hours after lunch today and tomorrow. So first, we just really quickly wanted to introduce ourselves, and I'm actually going to give you a brief overview of the Angelo Lab GitHub, how to navigate it, what information you can find there, and how you can use that to directly communicate any issues you're having with the computational team. Okay, so this is the Ambu Lab GitHub. So you can find all of our code there. The two repositories, like people have mentioned, that you really gonna want to look at are the Talk repository and the Art Analysis repository. So if you navigate into one of these, this is what it's gonna look like. If you want to look at the code, you can. If you really want to look at any open projects we have, you can look at pull requests. But the two things you're probably going to want to focus on are first, if you scroll to the bottom, there's going to be, like Noah mentioned, an in-depth README documentation. So this is going to give some explanations of what kind of code is here, why it's there, what it does, how to troubleshoot it if you have any issues. The two other really important things are an issues tab and a discussions tab. So to use either one of these, you're going to have to create a GitHub account. So it is a free account you just make on the website and sign up with your email. So this, yeah, sign up. So the discussions tab is going to be for more general questions about, like, how does this work? Maybe why do we do it? Any feature requests you have. So you can just start a new discussion, select category. Most likely you're going to want to use the Q&A category. But if you want to, you could just use or create a general discussion. That's fine. And then the really important thing is knowing how to submit an issue. So we have a bunch of open issues that we steadily work on, but we just ask that if you run into like a bug in the code or you get some errors that seem really scary, you can go ahead to the GitHub page, check the open issues to make sure it's not something we already know about and are actively trying to fix. And if it's not, you can just go ahead and submit a new issue. Like I said, most likely you're going to submit a bug report. And when you go into this, it's going to prompt you to give us a little bit of information about how you got the bug, maybe a screenshot of what the error looks like. And steps on how we can reproduce it, and so we can really diagnose what's going on and work on fixing it. And I skipped my slide, but my name is Cami. I did my undergrad at UCLA in applied mathematics and computer programming, and then I just received my master's in mathematics and statistics. I joined the lab in March of this year, so actually tomorrow 6 months since I started. And I've mostly just been working on creating maybe processing tools and really just cleaning up and updating the Italian pipeline is what I've done in the last like 3 months.
You're a movie enthusiast and a sports fan.
Yeah, I am. I'm also, yeah.
That was also.
If you want that tidbit of information, you can have it. And then I'll go ahead and let them.
I'm Alex. I'm also on the computational staff team. I have put together the tiling notebook. You'll also have the fortune or misfortune of hearing my voice.
Fortune.
Of hearing my voice during talking about the tiling tutorials and setting them apart. I've also worked in close conjunction with Candace and getting the infrastructure set up for pixel and cell clustering. And I've also just been helping out with building up the general infrastructure of arc analysis and talk in general. I got my bachelor's of science in computer science at UC Davis. Then went to Columbia for my master's in data science. I have a background in software engineering, natural language processing, and network analysis. Outside the lab, I'm a classical pianist. I have a few prior years of teaching, and I'm also a casual singer. Never taking formal lessons, but I think my voice is pretty good. I'm also an ultimate frisbee player. Love playing tennis, and I'm unofficially trained for a half marathon. I'll get there one day. And I'm also a major travel enthusiast. This picture is me at Mont Saint Michel, from a great place in Normandy. I've mostly been to places in Central America, Europe, and Asia. Hoping one of these days I can get to Africa, Australia, and South America.
I'm Sri. Currently, I develop image analysis tools using MIBI data, and then I improve the general software engineering pipelines that makes our lives a bit easier to actually develop. And I got my BA at Colorado Boulder, last year, and I just joined the lab in February, so it's been 7 months about. And yeah, outside the lab, I really like to cycle. That was before my bike got stolen, so now I can
Oh.
yeah, it's on the to-do list. And then besides that, I really like to do, I'm a 3D art, and I'm just been learning a lot about that.